Hey everybody, welcome back to Q&A with A&V. I am Vincent Racaniello, and sitting on the other side of the wall is Amy Rosenfeld. Hello, Amy.
Hello, Vincent. It's not exactly the other side of the wall. We don't have an annex today. It's more like
once,
on the other side of the moat.
Vancouver,
I refer to the
BC, Houston, Texas. Somebody said hi, Amy.
hello.
Is there anyone from Australia? Sweden? That's cool.
Yeah.
Okay, hello everybody. Cleveland, Ohio. Hey, that's not Australia. France. We're in France.
Look, you can't, you're going to have to deal with it. Yeah, Kosovo.
Wow, cool.
He seems to be a lot. He listens quite frequently.
Oh, yeah, you recognize Dr.
Well, I
recognize,
no, I just recognize somebody from Kosovo.
Okay,
the person in San Francisco can go say hello to my sister.
So I got Cyprus, we have Mexico, Norway, Boise, Ireland, 2 a.m. in the Kosovo, Trinidad and Tobago again. Hello, Trinidad and Tobago. Ohio, Questella, I recognize the sunglasses. Michigan, Houston, Ireland, Italy. This is pretty cool, Amy.
It's great.
Reading, Pennsylvania, Chinatown, New York City. Yay! You know Wohop?
I could do some dumplings.
17 Mott Street.
Hey, I'd like I like 5 Pell Street. Joe Shanghai, a little soup dumpling. Sounds good to
Wonder
me.
says, hi, Amy, you rock. Calgary, Alberta, Denver, New York City. Joseph is in New York City. Yep.
It's good.
Boston.
Are you just reading where people are? Like
for now, Kentucky.
Well, I'm kind of, I have a grant to write.
Joseph says, Amy, I'm in New York City and you're beautiful.
Thank you very much. But I have a grant to write. We need to speed this up.
Amy, you leave at 8:45, right? That's your thing? Maryland?
Yeah, 8:45, yeah. I have to work.
Looks like Moderna. Oh, let's do this one now. Here. It looks like Moderna has announced their vaccine produced 6-fold lower of antibody titer.
That is true. That's what the paper said. That's what Barney Graham's paper said. That's why they announced that they're making a booster variant.
So in other words, if you take serum from a vaccinated person and you try a neutralization assay with some of the variants, not all of them, you get a lower level. And Moderna says it's probably enough, but we're going to update the vaccine anyway. That's right. Right, Amy?
Yes. Yes.
See, Ace Wonder says, Amy, don't rush us. We love you.
Well, I have a limited amount of patience.
Look who's here. Kia Ora from Pongaroa. We have a guy on TWIP who says Kia Ora from Pongaroa.
He must be your TWIP listener.
I don't know, Andrew. You're the TWIP listener? I'm not sure.
I'll let you know.
Here, question for both of you. If you were offered Sputnik 5, would you take it given no alternative? So Sputnik is the 2 adeno vaccines that the Russians have developed, Amy. And one is different serotype from the other. If I had no alternative, yeah, I'd take it. Sure. Would you, Amy?
Sure. But I don't need you to refresh me. Like, I know what it is.
I'm just chatting, Amy. Chill.
Okay. Well,
Chill,
let's
Amy.
just streamline this.
Moderna is the best, as far as we know. Best vaccine. Well, Pfizer is pretty good as well. So I would say Moderna and Pfizer have the highest efficacy. What do you think, Amy?
Yeah, I think they're both. I think they're compatible. I don't think that there's really a difference. I mean, I got Pfizer and you got Moderna, and we're still standing. So that's all that matters.
Do you think COVID is now endemic and will never vanish? I think it is. What do you think, Amy?
Yes, I think it's never going to vanish. I think most viruses are endemic, actually.
SARS-1 is no longer, is it?
Probably not, but smallpox you had to eradicate.
Yeah.
Yeah. Well, we had.
right?
So it would be endemic if we didn't eradicate it, right?
Yeah.
We actively intervened. It's an act.
so,
We
yeah,
actively intervened.
yeah, it's
So
fine.
it's different.
But this one isn't even eradicable because they're animals that can be infected, non-human animals. So that's a problem. Frank wants to know how you're feeling after last week's booster. Are you okay?
I feel better than I did before, but I was very unhappy.
The problem with this week is Amy is writing a grant with 2 other centers, and they're giving her headaches. And so
No,
when that's over,
that's this week. Last week the booster made me ill.
yeah,
But this week, yes, this week the grant, the grant, and then the abstract for some meeting. It's amazing.
would it be possible the new variants have better replication capacity or fitness, low CT? If not, how much mutation would be required? So I think we don't know exactly how much more fit they are. Something's driving them through the population in UK. But I got to say, in California, they've been there since November, and they are not displacing the previous strains. So it's not a straightforward thing of being more transmissible. It's something else more subtle that we're not getting.
Well, he didn't say it was more transmissible. He said it was more fit, meaning it replicated more efficiently. That's different than transmissibility. But you clouded the issue, so you should correct it. So if you put the 2 in a tissue culture cell, and you wait 24 hours and you put 3 PFU of each in the cell, do you get more than 1 out? Or does both at the 24-hour mark, does both give you 10?
Yeah, has that been done?
Well, that would be Vinit's answer, right? That's what Vinit is doing.
Culture, people think they're slightly advantageous in tissue culture, but as Vinit said, he's not sure that's the right model. So,
but that's irrelevant. The question is, does it have better? Does it replicate better? Is it reproducing better?
In cells and culture, but does that translate into people? Is the question. We do not know, right?
Keep confounding the issue, but it's fine.
I need data on or updates about whether they're actually more transmissible or cause worse disease.
Now you can talk about transmissibility because the question addresses transmissibility.
No, there's no new data about transmissibility in humans. It's all indirect and associative, but and they don't seem to be causing worse disease. The most concerning issue are the whether they have reduced reactivity with convalescent sera or post-vaccination serum, which some of them seem to, and that's to me more concerning than anything else.
Yes, unfortunately, though, the Prime Minister of Britain is using the phrase "causes worse disease."
Why is he doing that?
Because he wants to justify locking down the country.
All right. Would human body cells infected by the mRNA nanoparticles be attacked by immune cells and eliminated? Possibly. Yeah, I talked about this last week. They could go into muscle cells, then there would be protein displayed on the surface, and then that would be attacked. So, yeah, but it would not be extensive because it's limited to your muscle. What is the primary efficacy endpoint used in influenza vaccine trial? It's not infection, it's disease. Now, I don't know if it's which of those 3, disease, severe disease, or hospitalization. I'm sorry, I don't know. May be compelled to take Coronavac. If there's nothing else, they say, what beer would you have? Some people say the beer that's in front of me. But Amy doesn't drink beer, so.
Well, that's irrelevant.
Oh, Andrew's the TWIP guy. Good. Nice to meet you, Andrew.
See, he listens to all your podcasts. He listens to Immune and TWIM and Tweevo and Twin. See, he's a great listener.
I think at the end he says Namia, right, Andrew? That's how you sign off. Anyway, I'm going to get Daniel and Dixon to live stream TWIP sometime.
Seriously?
Yeah, don't you think that would be fun?
Yeah, I just didn't see that Daniel nor Dixon would be into it, but okay.
I think Daniel would be.
Do you see Dixon?
He wouldn't know the difference. I wouldn't even tell him. Just do it. what, what do you think? Is that bad? Is that bad to do?
It's fine. It's fine.
Well, I have to say, you
He
guys, I
might.
said to Amy, I said, you want a live stream? And she said, sure. So
No,
that's
that's
what.
not exactly what I said, but that's okay.
What did you say? I forgot.
I said, you asked me if I would help you answer questions. I said, sure, I'll help you answer questions. One night a week, 30 minutes. Now it's 45 minutes long. And then you said, we'll make it live stream. And I said, okay, well, you're in charge. If we get 15,000 people who need to hear me every week live, and then the 15,000 people just went away. But that's
well. We do
okay.
600-700. That's amazing for a live stream.
I don't know anything about numbers.
So are we screwed with this new variant? Well, there are a bunch of
No.
variants. No, we're not screwed. We're good. The vaccine will be good. We'll change it if we have to. No worries.
Why would we be more screwed by a new variant of SARS-CoV-2 than we are by a variant of influenza?
Yes, exactly. I mean, one could argue that, yeah, I think that's absolutely key. We do this with flu all the time. We make new vaccines. No problem. Amy has the clarity. Okay, here's one from Jaspel. If you're fully vaccinated, catch a variant, will the immune system automatically adjust? Will you get slightly sick, but not enough? So you will make a memory to the virus you were immunized with, or the mRNA, and some of those antibodies will inhibit, but not fully, and you may get slightly sick. I think it's like flu. When there's an amino acid change, you get slightly sick, but not as sick as before. So, Amy, did you know that J&J is doing a 2-dose trial? I didn't know that.
Yes, I did.
And why are they doing that?
They had some logic that I don't remember. But no, it's not has to do with one dose was the efficacy of one dose was too low. No, it doesn't have to do with that. But I don't remember. I have a lot of stuff to keep in mind. This is not what I do.
I have to read this one from Amy Lipkin. This is amazing because Amy is collaborating with Ian Lipkin here at Columbia.
Maybe she's Ian's sister.
Is it normal for someone who's had their second vaccination to have no symptoms at all? Yeah.
Yes, I think Daniel actually really had no symptoms from his second vaccination. I think he just pretended that he like forgot he had some meeting on Friday, that second day after the booster, and he's like, "Ugh, brain fog." That's what he said last week on your Twiv with him.
And you take first dose Moderna and second Pfizer.
Actually, now you can.
Yeah, who said that you could? The FDA?
CDC. CDC said that you can.
Yeah, those two.
It's not highly recommended, but you can.
Nice. Hawaii. What do we think about Novavax? So the data for phase 1 and 2 looked pretty good. It's a protein-based vaccine. It's spike, and it aggregates to form some kind of particle, and it's got an adjuvant, and I hear it looks good, and the phase 3 is ongoing. So I think it could have promise. So we'll see. It's going to be another month or two, I think. Phase 1 of Novavax came out. I'm not sure about phase 2. I don't remember covering it on Twiv. Hello from Canada. There's evidence the effectiveness of vaccines may vary on different ethnic groups. So the mRNA vaccines were tested in different ethnic groups, and there was no difference. So I don't know exactly what you're referring to, Sharon. Do you know anything about that, Amy?
I don't know.
How do viral vector vaccines work? In our country, vaccines use adenovirus as vector for mRNA, the adeno infecting cells and making the cells translate. Yeah, the adeno goes in the cell, the DNA goes in the nucleus, it makes mRNA, the mRNA goes out in the cytoplasm and it makes protein and it makes spike. But the adenovirus depending on which one, is either replication defective or competent. Let's see. Can an adenovirus vector be reused? Well, I don't know what you mean by reused. Do you go back in the lab and use the same vector for something else? Sure. It's on a plasmid, so it doesn't matter. You just tell Amy, go get it out of the freezer and, and put the different protein gene into it, right?
No, you don't tell me nothing. No. I am not your technician.
Not me. It's some guy, some CEO in a company.
Yeah, well, I don't work for a company, so
I don't make vaccines. No. I'm wondering if there's a test to discover a person's susceptibility, or if it is even possible. So that's an interesting question, right, Amy?
Yeah, it's a biomarker test. And since you'd have to know what the biomarkers are and stuff, we don't really know what that is. I mean, the suggestion is that if you have these inborn errors in type 1 interferon pathway genes by Casanova, but it's not really clear that that has any real relevance and not just correlative, since it's not it doesn't stimulate the production of type 1 interferon. So it's not clear, unless those gene products have alternative roles, what that would mean. But it's something that could be looked at, and it could be interesting.
All right. So someone said my mic is too quiet, so I just turned up the volume. Let me know if that's good. Amy, what were your symptoms from vaccine booster? How long did they last?
So I had 101 to 102 fever and chills. And so the first day, I really didn't feel very well, but I was here on Wednesday. And then I would say about until Sunday night, I had night fevers. And then like about a week later, it ended. It's fine.
But Amy doesn't miss days because of illness. She once had pneumonia for a year and never missed a day of work, right?
That is true.
So I just want to show you that this David loves the Q&A, and he loves your insights, Amy, okay?
That's great.
All right, any idea what level we need to get to with the vaccine? Will it 2/3 of the population be enough?
Well, Fauci keeps raising the amount of herd immunity, right? It was started off at 60, 70%. Now he wants an 80 or 90%, right?
Yeah,
so that would mean approximately that amount of people would need to be vaccinated.
Yeah, I don't think it's that high. I'm not sure Tony is right, but I think if you just base it on the reproductive index, it should be 70%.
Yeah, but the reproductive index changes.
Well, the one that's calculated in the beginning before interventions, 2 to 3, 50 to 70%. We want to wager a little bit of something, Amy, like wine or
no,
something?
Because you never pay up. So no, I'm expensive, and when I win, I never get anything, because then I'm told I didn't really win. So no, I'm not. I don't need a wager.
Are any of the up and coming vaccines a one dose? Yeah, the J&J.
So J&J is one dose.
Yeah. I got an email from J&J scientists thanking me for my lectures today.
That's great.
And I said, I'm thanking you for the vaccine.
That's great. I think I saw in the journal that the stuff is supposed to be out next week or early February.
Good. How common is second shot blues? I don't know. I just have anecdotal data. Do you have any clue, Amy?
No, I'm not a clinician.
36 hours is about what Amy fell ill with, right, Amy?
Yeah, but then I had like night sweats for a few nights up until Sunday. So I would say like 5 days. It's fine, whatever.
China is doing mass rectal swab screenings because viruses in feces. Why have not public toilets with no lids been a source of infection? I think there's probably not enough virus in feces, right, Amy?
I would think that there's not. I'm not sure. So are they just looking at RNA or are they culturing live virus?
Just PCR.
Okay, so I mean, I can find very, very rarely, but we can find EV68 RNA, but it's not infectious virus, despite what others have tried to publish. They are wrong.
I think that very few people have found infectious SARS-CoV-2 in feces. Yeah.
Right.
Moderna study to assess whether the vaccine prevents transmission is stalled because of insufficient funding. Anyone else working on it? I thought Pfizer was too, but I don't know these inside things. Did you know it was stalled, Amy?
I had no idea.
Wow.
But I find it very hard to believe that the company is out of money.
Yeah.
That doesn't make any sense to me.
There it says, the Wall Street Journal, COVID vaccine study on preventing transmission is stalled.
Okay,
Yeah,
I missed it.
This is being done on college campuses. Yep.
I missed it.
They don't have enough money. And Amy, you read the Wall Street Journal too, because you're a
I
financial
do, but
person.
I'm not a financial person. I'm only a financial person when the chair of the department comes and says, "You're broke. Write another grant." That is when I'm a financial person.
If you join a phase 3 trial like Novavax and it turns out to be ineffective, can you then take an mRNA vaccine? Sure. No reason you couldn't, right, Amy?
Right.
No reason at all.
But you said one time that in order to do a new trial, you had to have the standard of care. So automatically there has to be an arm in that trial that then you just move over to getting the mRNA vaccine.
Yeah, right. For those who believe that the variants are more transmissible, what evidence are they basing their conclusion on? Okay, so several things. First, that in the UK, the variant displaced the previous strain. It seemed to be associated with higher levels of RNA in nasal swabs by PCR and RNA-seq. But as Amy will tell you, that's a single time point. And how do you know you got that person at a time point that's relevant? That's it. So they make statistical models of these data and they say, looks like it's more transmissible, but the models can't be used to conclude that. You still can't conclude that D614G is more transmissible in people. It's still a hypothesis.
I'm unclear how one extrapolates having more RNA as being more transmissible. I could just argue that it is that replication is more efficient or something.
Yeah,
and you're actually making more defective particles. So I don't know that I don't know that I would go with that. But hey, that's what they want with.
Yeah, that's what they're doing. I agree with Amy. What about the Houston, Texas variant? You know, there are various variants popping up all over the place. One, because we're starting to do more genomic sequencing, right?
Yeah, I believe that has been, I think that is one of the, 7 points that Biden made?
Yeah.
In the document you read last week on Twitter?
Yeah, more genomic surveillance. And, you know, the UK variant has been in the US since before it was picked up in the UK. So, you know, finding new ones is not really a big deal in my view. Has anybody performed PCR Southern blot? I don't know what you mean by that. To check for better transmission. PCR wouldn't tell you about better transmission.
Well, Southern blot won't either, too, because it's for DNA and this is an RNA virus.
Well, you're amplifying the, right, with PCR. So but I don't know why you would Southern blot it. You can just do the CT and calculate how much is there if you want to do that, right?
Yeah, I don't understand the question. I don't even understand the technique.
With the many mutations, you think we'll have to continually get vaccinated every 6 months? I don't think so. I think, well, it's not clear that these will continually arise, and it may be a year, as Amy said last week, a couple of years, right? That's your shot, correct?
Yeah, yeah. I mean, I think like you made a vaccine and you found out that in actuality it needed to be like multivalent. kind of like polio. They made a vaccine. Then they found out that they forgot you had 2 other serotypes. You needed to include them.
Yeah.
And voila. So at some point you'll have a multivalent vaccine and you'll get vaccinated once every 5 years.
So Wild Bunny is going to sleep, but she wanted to say she's a big fan of Amy. She's so amazing, forthright, and honest.
Oh, thank you. I'm sorry that it's so late for her.
And Amy, also Wild Bunny likes my course. Thank
It's
you.
great.
Good night, Wild Bunny. Okay, Lucky Sixes says for influenza vaccine, the endpoint is actually infection. That's what I gathered from watching ACIP meetings. Thank you. Will infection by one variant give you some protection from another? Yeah, you will get cross protection. How much depends on how much cross reactivity, but I would think that you would get a mild disease, just like you do with flu in the yearly antigenic drift. Thank you, Amy. You just said what I've been saying all week. Boris Johnson's claim that it may be more deadly was an attempt to scare people.
The endpoint of the vaccine is actually clinical benefit.
For flu?
Prevention of influenza illness and its complications. So I don't know that that is infection.
Okay. That's correct. I took an antibody home test for the Royal Imperial College. I waited 10 to 15 minutes. It showed a red bar, and then 6 hours later, it had 3 red bars. So there are always 3. There's the control. No, there's 2 on mine. Sorry, the control and the other red bar, which is positive if you have antibodies. So they should be marked on the cassette, C for control and then the other one. So, but it should develop in 10 to 15 minutes. So I don't know what 6 hours is. I think you should in the 10 to 15. Go ahead.
6 hours is like the bleed through. Like you can take a pregnancy test and it says wait 3 minutes and you see that you're negative, and you come back 6 hours later, it'll look like you're positive because it's the bleed through.
Right. So don't wait 6 hours. Yeah. Mark. Oh, that's the same one. Sorry. What percentage of viruses are good for us? What do you think, Amy? Do you have a number?
You're the one who gives the class. It's the end of the first lecture. Come on. You just gave it like 3 weeks ago or less. You're only on lecture 5.
99% of viruses are good for us. That's just amazing. I'm really cranking up my volume here. Now it's redlining on my meters, folks, so that's bad. But if it's okay with you, has there been any information about getting other vaccines, such as Hep B and COVID? Would there be any problems? Do you know anything about that, Amy?
No, I think that they tell you to get other vaccines, just not on the same day.
Yes.
But I think that that is true for all vaccines, is that you're not supposed, like, I think that they are now starting to frown upon the fact that kids get DPT and polio on the same day.
Can you please explain aerosol transmission and if this is the main route for SARS-CoV-2? I would call it droplet transmission because when you breathe or sneeze or cough, you make large droplets and then very small aerosols that go a long way. So that's how I view it. And I would say that 90% of the transmission is by droplets that fall to the ground within 3 to 5 feet. And there's a small percentage of aerosols, we think, but I don't know how much that is. I asked Tony Fauci a couple of months ago. He said he didn't know. And, Christian Drosten recently said 10% by contact, surface contact. What does this mean? Just a couple of hours. Come on, Amy. We love you. What, a couple of hours for what?
I guess the live stream. A couple of hours. When, let's look at this. I've been up since 5. I ran. I've been here since 10. I've done experiments. I wrote an abstract to a meeting or something or other that I'm not going to. Did more experiments. I've yet to go home and eat dinner. And it will be 9 o'clock before I get home. And at some point, you need to eat dinner. So let's say that's 9:30, 10 o'clock. So that makes it 1 p.m. for me, 1 a.m. for me to go to sleep and get up 4 hours later.
Cheryl wants to know, why do some people have side effects from COVID vaccine? Others feel like they have their butt kicked. Would it relate to how sick they would have gotten if they got infected?
No relationship to viral infection.
No.
Whatsoever. It's just the way your body responds to different things. Not everybody gets the same disease, even if you got the same bacterial infection or whatever.
All right, UK Nerve Tag, which is that committee putting out information on these variants, concludes realistic possibility that infection with VOCB117 is associated with an increased risk of death. So I don't like any of the conclusions of Nerve Tag from the beginning. I don't agree with them. This variant is in California, and it's not associated with increased transmission. It's not associated with increased death. You just interpret that the way you want. I mean, it's just not blanket more lethal, more transmissible. John Campbell. I don't care what John Campbell reports, okay? He's just reporting what Nerve Tag says and doesn't look at it critically, I think. Is it possible that with everybody using masks, the new variants are ones that are attached to smaller droplets? Small droplets are caught by the masks, aren't they, Amy?
Yeah, it depends on the filter. It depends on the mask and the filter size. Plus, like an N95 has an electrical charge. So it all depends on what kind of mask you're wearing.
Oh, this one's for Amy. What are your thoughts on colchicine for treating COVID? You told me about this the other day, right?
Yeah, Andrew emailed me an article that said some group in Montreal found this. The drug for treating gout. It's an anti-inflammatory, right?
Yes.
It dampens the inflammation.
Yeah, that makes sense, right?
Mm-hmm.
What about double masking? Dr. Fauci is now recommending this. Why not? Not sure you need it, but if you're wearing masks poorly, maybe it would help. What do you think about double masking, Amy? No.
Well, if you're wearing one mask poorly, I'm not sure that you're going to wear 2 masks properly, but okay. I guess it really all depends. Like if you're wearing a cloth mask that's pores are larger than a surgical mask, then I guess it makes sense. If you're wearing an N95 whose pores are the smallest and it has an electrical charge, maybe it doesn't make any sense. I mean, 2 N95s, I'm not sure is really going to make a big difference. But, you'd have to like qualify this somehow.
All right, a few comments on John Campbell. He's a nurse known for over-the-top hysterics, okay? And then he is too trusting in what the UK government says. So John Campbell says, this is what the report says, but he can't look into the science. So he says, the report says they're shedding more RNA, but he can't make a judgment about that because he doesn't understand that's not infectious virus. So I
trust you. I've never listened to him. I have stuff to do.
I read somewhere that there are tens of thousands of viruses that can jump to humans. Probably more. Tens of thousands in one bat. So there are lots of bats in the world, and they're all full of viruses. They could they are candidates for spillover into humans, and they are 20% of mammals. Mice, too, they're very numerous. They have a lot of viruses that, and some of them spill over and cause disease. Yeah, they're plenty. People need to be like Amy. She works in the lab all day. She can't have any spillover, right?
No, but I have a different agenda than that.
Vaccination in Israel seems to be doing okay. Is that correct, Amy?
Yeah, they seem to be, well, they seem to be almost, I think, a third of the way through their population. They anticipated that they would be fully vaccinated, I think, one round by the end of March.
Excellent.
It's quite the tour de force.
Are both of you getting the vaccine? Amy has got both shots already, right?
Yes.
And I have one so far. I have the next on the 16th. I have no doubt that it's the thing to do. I'm curious how much hours you spend a week podcasting and podcast prep. I'm not going to say it in front of Amy. She'll get mad at me. No, I'm just kidding. It's a joke.
I know. I'm interested in how you're going to factor my participation in this.
I do a lot because now we have TWIV 3 times a week, and now I'm teaching. So that's 2 lectures a week that I record and post. And I do all
that's
the
not
editing.
Podcast, though. But that's not a podcast.
But I do 4 or 5 podcasts a week, and what's the matter? 3 TWIVs, right? And then one other, let's say 4. Is that realistic, Amy?
Yeah.
But the good news is we might get a studio, right, Amy?
Yes. We're going to go look. I have it all set up for February something after my grant deadline.
I know I didn't answer that, Max, but I'd really have to calculate it. A lot. Abraham wants to know, is there an AI system that tests every mutation to see if it affects binding? Yeah, there are. I'm not sure how good they are. Because we did a paper where they looked at all the ACE2 spike interactions, and they made mistakes, right, Amy?
Yeah, we don't. Computational experiments are overrated. You throw in some fancy words and people think that you're actually doing something important.
Is it possible to invent a scratch test for CoV-2 personnel susceptibility? Not at this time. If you knew what mutation influenced susceptibility, you could, but we don't know at the moment. And we're going to be more. So apparently Israel bought millions of Pfizer and Moderna doses. Okay. Is it possible to have a career in virology with just 2 masters? Well, it depends what you want to do, for sure. I know people that have masters and they work in laboratories and they're absolutely happy to do that. Wouldn't you say, Amy?
Yeah, but I also know people who have master's degrees and work for pharmaceutical companies and don't work in labs. Sometimes they're higher than the PhDs.
We were running out of vaccine in Alberta. People's second dose is being delayed. Any thoughts if delay is too long, how much that could reduce efficacy? Well, I don't think it's going to reduce the efficacy. The problem is you could get infected the longer you don't get the second dose, right? You're increasing your chances of getting infected. And we don't really know how much the first dose protects you, right, Amy?
Well, I wouldn't have phrased it the way you did, because I don't think that that's an accurate phrasing, because it only tells you one half of the story. So you're focused, if you don't get a first dose or the second dose within X amount of time, the probability of you being infected is greater, which is true. But if you got the first vaccine, you don't know when antibodies actually max out, when the highest titer is, or how long that is maintained. And some of those graphs from Theodora and Kramer and other people, they seem to be very long time things, months. So, you know,
Yeah.
you have a lot of protection by that at that point, and we're only counting antibodies. You're not even factoring in T cells. So to say to say the probability of you getting infected is greater than, you really don't have any idea.
Well, the more time
It's
you
like,
wait, it's greater, right? You have more of a
well,
target.
I don't know. Well, is that really true? Or do I just allow the T cell response to kick in and I'm equally protected?
It's a time thing. You know that. The more time you're unprotected, the more
Well,
likely
you're
it is to
not.
get.
But see, the thing is you're saying you're unprotected. You're not unprotected.
We don't know how much you are protected. That's fair to say.
So then don't start off with you're unprotected. That's not
I
correct.
didn't start off. I just said the longer you go,
you have
the
no
more
idea.
likelihood you are to get infected, and we don't know how much protection the first dose gives. I think that's appropriately couched.
No, I don't.
Okay,
But
you can agree to disagree, Dr. Rosenfeld.
and they're running out because Pfizer has a manufacturing problem, and there's no plants in Canada to do it. And Andrew has sent me emails about this, or texts.
So apparently in Germany, BioNTech is retooling their production to scale up, and there's a momentary halt. That's what my German guests told me today.
Could be.
Where do you have time to read all these papers, Amy?
I don't know. I don't ask those kind of questions.
Get Dixon a nice mic like Amy's. I have offered. He doesn't want a mic like Amy's. He wants a headset. So there's not much I can do about that. I'm sorry that you lost your wife, Dev. I'm really sorry, and I'm glad the content here has helped you to cope. It's terrible. My micro student does not want the vaccine due to it causes infertility.
How does it cause infertility?
I have heard nothing on that. That's a anti-vax excuse to not take vaccines. There's no evidence that it causes infertility.
I'm unclear how that would even work.
Right. Is that right? Sanofi's going to help with Pfizer production?
I don't know. You know people at Pfizer. I don't know.
Pfizer.
Whatever.
Pfizer. All these production things, I don't know anything about it.
I don't know. I just think that you had 10 months. You needed to get it together.
So we're going to get you over 100,000 YouTubers. Thank you, Questella. It's 81,000. It's very nice. I appreciate it. And you guys can all give a thumbs up tonight, too. Push that up. It's at 78. What about when you want to take the vaccine, but it won't work because your thymus was taken from you? Yeah. So you have to get monoclonals then in the case of an infection, right, Amy?
I guess so.
Without a challenge, how do we know the vaccinated group has the same exposure?
I'm sure that there's a statistical calculation.
Yeah, that's why you have to have a lot of people in both groups, 15,000, 20,000, and they're all in, many of them are in similar areas, so you can guarantee that they're having similar exposure. That's right. It's all about the power of the trial. Amy, when do you want to go? Now? Do you want to take a few more?
I'll take a few more and then I'll leave. Too many people in the hallway.
You want to close the door?
No, it's okay. They're all going out.
You know anything about getting vaccinated if you have MS?
I don't know. You have to talk to Daniel. I'm not a clinician. I I don't ever want any responsibility. So.
What happened to the UK challenge trials? What happened is they have a lot of cases. They don't need a challenge trial. That's it. Any word out of China as to origins? No, there's a WHO committee that's going over there to sort that out, but it's going to take them a while.
Oh, you mean they aren't just going to go to the bat cave and ask the bat?
Right. Exactly. Oh, look, they pushed
Here,
us over.
let me interview you. How's that?
Over,
Can we interview you?
Sure. They put the bat up to their ear and they ask if the virus is there.
Really? I thought that they took a mic and they asked where they were from, how they were feeling today, what they had for breakfast, and then,
oh, yes, got it. In YouTube's, ask us to like the live stream by clicking on the thumb below. Yeah, I know. Just give us a thumbs up. Thank you. It went over 131. Did you take NSAIDs after your second shot?
No.
The infertility claim is based on a small sequence in the spike as syncytian. Yeah, right. That's it, Amy. You remember that? But there's no basis for that.
Oh, yeah. I think somebody, I don't read those papers.
Yeah, that wasn't very good. This Laura's in the Bay Area, and she wishes she could bring you healthy dinners. When I visit, I may show up with a care package.
It's very nice. She can send it with my sister. My sister is coming soon.
Hey, look, this is interesting. If I get a dose of vaccine, then I get COVID, do I still have to take the second dose? I don't know. There's no guidance for that. I would say there's no reason not to take it. It'd be a boost after you get better, of course. Mm-hmm. Oh, so Christella said, get Batman to ask the bats, what virus.
Yeah, Lympha and Peter. I got it. It's cute.
Here we go.
Right, 3 more questions, and then I'm done.
The Orphate reading frame is missing, right?
Mm-hmm.
This is a sign the virus may be on a pathway to weakening. That's right, Ephraim, and nobody's talking about that, and that could contribute to transmission. I have no spleen. Is that a problem with the vaccine? You have other lymph organs, right? So you're okay.
My stepbrother has no spleen. He's getting vaccinated.
Friend who monitors China has told me COVID is out of control in 4 cities and people are collapsing from the virus. Any comments? I don't think it's out of control. They have it in. They're really good at controlling it. They have cases for sure, but I don't believe that they're out of control.
No idea.
Oh, here's one for you, Amy. Maybe this will be your last.
Okay, this is my last question.
Is there research showing that SARS-CoV-2 is a neurotropic virus and has the potential to persist or remain latent in the CNS?
No. So I have to be nice. I really do, because it's a shit show. So there is a manuscript that suggests that when you open up the freezer and you take out brain organoids, you can infect them. Put the brain organoids back in the freezer. So what is the relevance of that experiment? So a brain organoid generally is at most 3 at the end of the second trimester of development. It does not technically have electroconductivity, and it is missing a ton of cells, in addition to the fact that the microglial, which are the immune cells of the brain, have not entered because they are from a different tissue. Mm-hmm. So, and you are not fully terminally differentiated. So that bunch of data is not applicable and has no biological significance. And then there's that paper from Nature Medicine where they looked at the autopsies, and I think that they saw very little virus in the actual olfactory bulb, like one person, but it was all in the olfactory mucosal, which is intricately connected with the axons of those neurons. Isn't that correct?
Absolutely.
And then they conclude that because it's autopsied and some of the tissue is not intact properly, that it might be they're hesitant to say that it is actually replicating within the neurons. And then another woman did a paper with organoids that made cerebral spinal fluid, so at least they're more reliable than the other organoids. And she does not see any virus in the neurons. She does see that it can potentially cross the choroid plexus, but it's from immune cells. And I believe that Ken Tyler has a review that also talks about this, that the neurological symptoms are due to the consequence of infection. And then the Hopkins groups correlated that, right? You would say that Carlos's abstract correlated that. Mm-hmm. And then there is the paper by Krickstein that talks about astrocytes, which I haven't gone through fully. So there is a lot of neurological side effects to viral infection, but it is not replicating within cells of the nervous system or the central nervous system.
All right.
I think that was a good summary, right?
It's a good summary. Thank you very much, Amy.
No, I had to be nice. Now I have to go home and have a drink.
All right. Good night, Amy. Thank you for joining us.
I'll see you tomorrow.
Yep, I'll be in.
All right.
Bye. Okay, folks. You got me now. Let me go to Ephraim here. As other coronas have shown this change before weakening. So that's the orphate loss. So it was seen in SARS-1 in 2003. It was a variant that arose halfway through and predominated the rest of the outbreak. And there's a hypothesis by Christian Drosten that it actually makes the virus weaker, less virulent. Not proven, though. Amy thinks the orphate is a hotspot for recombination, and the polymerase just makes mistakes there, and that's why it gets deleted. And Ephraim says, is there a risk that you would mess up other open reading frames? Probably not, because those are really essential, and I don't think the virus would be infectious without it. Can I ask about bats? Ebola, Marburg, SARS-1, 2, and others. Why do so many zoonoses spill over from bats, considering there isn't that much human-bat interaction? Actually, there's a lot of human-bat interaction. Many people get rabies from bats, and in China, there's a lot in the countryside. So, yes, there's a lot of interaction. That bats are 20% of mammals. So, yes, that's why there's a problem. And why they have so many viruses, that's a good question. They have immune systems that can handle them, and people are studying bat immune systems to figure that out. But we don't know the answer. And that is one reason. But not just bats are spillover risks. Rodents are spillover risks. Of course, for influenza, water birds are spillover risks, right? Real problem. There's a podcast channel called Dark Horse that claim the mRNA vaccines will predispose us to developing autoimmune disease. I don't see why that would be. I see no mechanism for mRNA making a spike protein for a couple of days to give you autoimmune disease. Not at all. What is Biden going to do with testing? Will it be like Mrna? I hope so. I hope they incorporate a lick a stick, a rapid antigen test that's cheap. I don't know the details. I just know that they want to ramp up testing and make it more extensive and so forth, but exactly what kinds of tests. But I would hope rapid antigen tests would be part of it for sure. For sure. What's the likelihood of the current mRNA vaccine to be effective against the identified strains? So first of all, you should understand there are variants being identified all over the place. There are variants in the UK, South Africa, Brazil. They've been detected in the U.S. Now in the U.S., some of them are different. So as we're looking, we're finding more. Is the mRNA vaccine effective? So they have only tested a handful. A paper came out yesterday saying the mRNA vaccine, the Moderna vaccine, is good against all of them. Slightly reduced blocking with one of them, all right? But they said it was probably still enough to prevent disease. Yet they're going to modify the vaccine so they can change it slightly to accommodate the variant. So that's the beauty of the platform. They can change it in about 6 weeks, and it should be fine. So I have no worries about that at all. I think we can respond with a new vaccine if we need to. My son is 8 and asks if there's any place on Earth that did not get SARS-CoV-2. I don't know. Awfully many countries did, but there may have been someone somewhere that didn't get it. If anyone knows, let us know. Any idea of vape-related pulmonary disease that was rampant in 2019? Yeah, that was a thing last year, and it was a concern. But I haven't heard anything. I haven't seen any clinical reports so far, which is quite interesting. If you got your second vaccine when you had COVID, would it worsen your infection? It should not. I don't think it would have any effect on the infection. Even Antarctica finally got a COVID case, so I think that leaves the International Space Station. Yeah, but it's not really a country, right? Are muscle fibers really APCs? No, they're not APCs, but they can present proteins on the surface, right, in MHC molecules, and that's how they would be killed by a cytotoxic T lymphocyte. So they do have an antigen-presenting system, for sure. It's lecture 11 or 10 in my course. If someone wore 2 non-N95 masks, would the friction create enough static to provide better protection? No, static is not going to help you. No. I don't even know if it would make static, but it's not going to help. What role does mRNA play in the immunity developing process? So the mRNA, of course, codes for the spike protein. It's delivered to cells, and the cells take it up, and they make the protein. The protein is displayed on their surface, and then they're recognized by the immune system. So it's essential for starting immunity. Good night, Pam. I'm glad you like it. Over-the-counter nasal spray that may work. I have not heard of it. If it's over-the-counter, it's not going to have an antiviral drug in it, and it's not going to be well tested. I haven't heard anything about it. I would be very suspicious. You're going to waste your money. If the virus is real, why does not everyone in households get COVID? Because everyone responds differently to viral infections. When you affect 100 people with polio, only one of them gets paralysis. But polio exists. Do you ever say polio doesn't exist because of that? So every virus has a different effect in different people. What would electricity do to a coronavirus? I have no idea. It depends the voltage. Hey, here's the thing. If it's a plus-strand RNA virus, would electricity do something? Ha ha. What has history shown us about putting politics in science or science in politics? Well, unfortunately, they're inextricably linked, right? Because science is funded by a political process. Congress approves the funds. So you can't separate the two. And they can't exist without each other. So unfortunately, they are stuck together. Yeah, you should bring Bob Fullylove. Man, he's great. He is great. 20% of all mammals are bats. That's right. High school teacher from India. What resources can I read so I can explain evolutionary origins of viruses? Send me an email. I'll send you some links to what you can do. Okay? Now, is there any data on the efficacy of the mRNA vaccine with delayed second dose? No, because they only looked for 7 days after one dose. So we don't know. Although maybe someone's going to get it. The easiest and fastest kind of test if you just need one to travel. So there are 15-minute PCR tests in many cities. You can just walk in. In New York, there's places called CityMD. And your insurance might pay for it, and you can just get it there. That's what I would do. Are you concerned that a bad actor would bioengineer a virus? No, I don't think they would know how to do it. Anything you do to a virus to mess with it, and I've been doing this for 40 years. I've been messing with viruses, engineering viruses for 40 years. Whatever you do makes them worse off. So I am not worried. I should reach out to Sean Carroll. Hello, Eric. Caltech, who has a podcast called Mindscape. Yeah, we just mentioned that on Tweevo. You want me to go talk about viruses and podcasting? Sure, I could reach out to him. I hate to promote myself like that, right? So you guys should reach out. Tell him to get me on. Did I read it that cats and dogs can get COVID? That's right. Now, the dogs are less susceptible than cats. So domestic cats and cats in the zoo, they get it, yeah. A lot of animals are getting it. Gorillas, the San Diego Zoo, gorillas got it. Minks, rodents. That's why it's here to stay in part. I'm way behind in the questions here, right? Is Merck working on the antiviral you're talking about? Yes, so they are working on this antiviral that we covered on TWIV, which was shown to prevent transmission among ferrets. It's a nucleoside analog. I think it's very promising, actually. It's called NHC, that Merck gave it a different name. So that looks good. It's an orally available antiviral, so that would be great if they could develop that. Any news about any other vaccine? J&J results are coming soon. Novavax is in progress. Merck, 2 vaccines, they decided to can them, unfortunately. That is unfortunate. Do any viruses besides rabies alter host behavior? You mean human or other? So there are plenty of non-human viruses that alter behavior. Among insect viruses, there are some cool things that insect viruses do. They for example, they'll infect an ant, and it makes the ant go to the top of a plant where it would never go, and then it gets eaten by a bird, and the bird then brings the virus somewhere else. Stuff like that happens all the time. In people, harder to know because you can't do experiments. Have there been any cases of reinfection? Yeah, there have been. Not a lot, but they're growing in number. And CIDRAP, the website, you can search for that. They had a list a few weeks ago of all the reinfections. That was pretty good. And it's hard to prove it because you have to get virus genome sequence from the first and the second and show that they're distinct viruses from a reinfection, whereas it wasn't just your virus stuck with you for a while and came back, right? So the bar is pretty high to show reinfections. So it's very, we don't have a lot of it. How can a vaccine stop symptoms but not reduce transmission? Well, it could reduce. Well, let me just give you an example. Polio vaccine, the one that you get injected in your arm with, prevents polio, but you can still get infected in the gut and shed virus and transmit it to other people. That's been well documented. And there are a number of other vaccines like that. So you can get infected and shed, but you don't get disease. Because the antibodies or whatever the immunity is protects the vulnerable site from disease, even when there's virus there. Ibuprofen affect vaccination efficacy? I asked Daniel Griffin this, and he said no. No. How would people with autoimmune disease react to an attenuated vaccine? I I think autoimmune is not the issue. Compromised immunity, right? People without immune systems, it would be a problem because we know if you give them polio vaccine, attenuated polio vaccine, it can reproduce in them for years. But I'm not sure that autoimmunity is an issue. The city MD line is long. Yeah. Well, there are a bunch of them in the city. Maybe you got to find the right time to go. No easy answers, right? Could these mRNA-based vaccines for the basis for future research? Yeah, for sure. Other vaccines, flu, HIV, HCMV are being looked at. And also for delivering proteins, therapeutic proteins. And I understand an MS therapy is being developed as mRNA. So yeah, I think it's very exciting. We don't have efficacy yet for Novavax. That's still ongoing, the phase 3. Which courses would you suggest having taken before taking your virology course online? Well, I would say you need to know some biology, right? Because I use a lot of terms. I throw out terms, and you have to know what they are. So just a basic biology course, which you can find online or at Khan Academy. Just take all the Khan videos on biology. You should be good. I got the Novavax first dose of study, so 2/3 chance of vaccine, 1/3 saline, and I didn't feel the injection. But you don't know. You could be one of these people that don't react, so you really can't tell. You could have gotten the vaccine. Why do viruses need a complement RNA? Oh, because take a plus RNA virus, like corona or polio. So the RNA goes in the cell. It's translated, right? You make viral proteins. But then if you want to make more viruses, you have to make more plus strands. And to do that, you need to make a minus strand. And so all viruses, you have to make the complement to make more of the strand that goes in the particle. What percentage of the population? From my calculation, 50 to 70% needs to be vaccinated based on a transmission index, the R value of 2 to 3. Now, Tony Fauci says higher, and he's obviously smarter than me, so listen to Tony. You reached out to, I'm sorry, but numbers is important also. But I can, why don't you send me his email, Eric, and I'll reach out to him. I've reached out to other people saying you should have me on to explain this, and no, but people didn't really respond. Antivirus specific TWIV with Tony Schounts. Yeah, I want to know about his see if his deer mice in the wild are susceptible. Can a person's reactions to flu take the COVID-19? They're going to say not to. So you have to tell, when you go to get the vaccine, they ask you that. Because I went 2 weeks ago and they said, have you ever had a reaction to another vaccine? And if you say yes, they won't let you get it. Are we going to get Jason McClellan on TWIV? He's going to be on Friday. You say jump, I say how high. How can herd immunity develop today if we can still transmit? That's a great question. If you can still transmit it, you'll never get herd immunity. But I think it's going to knock down transmission. I'm pretty sure of that. We just haven't shown it yet, okay? But I'm pretty sure it will. And I love it. I know it's herd, but I kind of love that it's herb anyway. You're wrong there. Politics is supposed to only discuss where the money goes. Yeah, I agree with you. They should follow science, but as you saw in the U.S., they don't always. And that's unfortunate. Nothing I can do about it. Would you enter a rabies vaccine study? You get $3,000 participation. It depends what they're doing. So sometimes they give you an activated rabies vaccine, and then they take your serum, which they then sell as convalescent serum. So if it's a vaccine trial, find out what kind of vaccine it is and how it was made and so forth. And then you can make an intelligent decision. If you want, you can email me, and I'll let you know what I think. thimerosal has been extensively studied, and there were countries that never used thimerosal using vaccines, and they still had increases in autism. So lots of trials were done. Millions and millions of people were looked at to show. And it's only now in the if you're concerned, it's in only the multi-dose vials of flu vaccine. Are they still using IL-6? So IL-6 antagonists or receptor blockers alone were not terribly effective, but in combination with other anti-inflammatories, they seem to be. And proning really helps. It helps get extra oxygen in the lung. Instead of lying on your back, lying on your belly. That's part of standard care in many hospitals now. And I'm not a doctor, but that's what Daniel Griffin told me. Seven countries without coronavirus. North Korea, I don't know about North Korea. Turkmenistan, Kiribati, Tonga, Palau, Tuvalu, and Nauru. I still have the domain twiv.tv, and TV is the Tuvalu country top-level domain. I guess you call that the .tv, right? I don't think a variant has caused any issue. I think they're just having a lot of infections for a variety of reasons that are not related to variants. I think we're blaming variants when we don't know what to blame, but so we blame the variant, and it hasn't been shown. Has PEG been shown to be the culprit? So Daniel Griffin said last time, yes, that PEG seems to be an issue with some of the allergic reactions. So if you have prior exposure and you're okay, then that shouldn't be an issue. What's my opinion on the transmissibility and fatality rate? So I haven't seen anything convincing about them being more lethal. Absolutely not. And the transmission, in my opinion, is only based on data that is not sufficient to prove transmission. I think these viruses are probably more fit, which means they do better and possibly in a variety of ways in the host, and that's why they're spreading. But to blame it on transmission, I think is not appropriate. And there are many virologists who agree with me, by the way. Are there other good things that use the same mechanism to enter a cell the way that coronavirus does? Yes, for sure. So the coronavirus binds a receptor on the cell via its spike, and it gets taken up into a vesicle in a process called endocytosis, and cells use that to take up nutrients that they need. So the virus is just kind of hitchhiking onto that, for sure. Will we revisit rhinovirus vaccines using multi-strain, multi-transcript RNA vaccines? Yeah, I think that would be awesome. We knew that we now know we can make, so there are hundreds of rhinoviruses, right? And this is the problem with, we thought always that we couldn't put so many proteins together, but now we know we can. And so now with mRNA vaccines, we could do that. So I'm very excited for mRNA vaccines and their potential. So give us a thumbs up if you like this live stream. And we're at 186. See if we can get over 200. I know he means well, and he knows a lot more about the virus than I know, but stating the fourth wave will be worse with the new variant, I don't believe there's any evidence that it will be worse. I think if there's a fourth wave, it's because there was going to be one because of susceptible people and transmission and so forth. But if we hadn't had variants, it would be the same with the old virus. And so, as I said, many people disagree with me. Many people agree with me. So there's not a consensus. Thank you. We got over 200. You guys are awesome. By the way, did I ever tell you were awesome? I really appreciate that you're here. Well, how many? 550 of you. Thank you. Even those of you who gave me a thumb down, thank you. Where I live, we only have access to Sinopharm and Sputnik. So if I had a choice, I'd take Sinopharm because it's two different adenoviruses that won't interfere with each other. And, even though the phase 1-2 data were small, I think it looked good. Many studies have linked increased deforestation to an increased risk of zoonosis. How do you think the effects of globalization will change the frequency of pandemics? So the problem is that we continually encroach on animal habitats, right? And that increases spillover. And these began, well, actually, the spillovers have always happened because humans have always interacted with animals. You know, we used to go out and hunt them. But now we encroach on their territory, so it's going to get worse, absolutely worse. And we need to have broadly acting antivirals and vaccines for them. What is Daniel's response based on? His clinical experience, I think. And Amy didn't take them because she probably didn't feel that she needed them. MIT biology course. Good. There you go. If you take that, you can be all set to take my virology course. Why is the U.S. delaying the Astrazeneca vaccine? So I don't really know the answer, but Daniel Griffin told me that they're having negotiation issues with the FDA and they can't come to an agreement. So Daniel actually thinks it's not going to be licensed, but who knows, right? What part does cholesterol play in SARS-CoV-2 infection? So cholesterol is needed for the uptake of the particle by endocytosis. So it's needed for entry of a number of viruses. So Carrie Mullis thought Fauci was a fraud. Okay, well, I don't think he's a fraud. I think some things you might not agree with and some things you might agree with. He's not a fraud. He's really interested in helping people. You know, the reason we have this Moderna vaccine is him. He got them interested. He said, well, maybe they were interested at Moderna, but he said, we'll pay for it. And I think that was really important. Well, how do I manage my time between communication and research? Well, I have Amy to do the lab. It's a joint lab, right? And more her lab now than mine, actually. It's just I've been there for 40 years in that space, but she is taking care of it. She's doing the experiments. She's figuring out what to do, and that frees me up to do science communication. And yeah, the pandemic, I've been doing more communication. So we increased TWIV to twice a week, basically. And I also get lots of requests for interviews, right? I talk to people, whoever asks me. I talk to groups. I get groups asking me to talk to them over Zoom all the time. I'm happy to do it. So I spend most of my day talking to people. Can a cat be infected with COVID-2 and transmit? That's a good question. I'm not aware of any example. It may just be that there aren't enough examples. But, you know, the minks in Europe, they get infected and they can spread it back to people for sure. So I don't see why not with cat. How much longer do you think we will have to have this negative impact? I think the fall of this year will start to be normal. I suspect we can go back to school normally in the fall. Most people will be immunized. That's Biden's goal, right? Kids won't be, but I think it's going to be okay in the fall. That's my guess. And if I'm wrong, you can call me out. That's fine. You're welcome. I'm glad you like the virology course. You know, here on YouTube, I've posted the first 5 lectures, or after I'm done with this, I'm going to post the 5th lecture. So, yeah, I do. Dr. Griffin did not adjust ivermectin. I agree. I think that sometimes he doesn't address things. And, you know, he's a kind of busy guy, and he's saving lives. And he, I think people write his summaries for him, you know, his assistants. So I agree there's something more. I'm not sure. Yes, I don't think it's an antiviral. It's doing something else most likely. And I'm sure we'll get to it at some point. I try to be open. I never say everything is known. Which vaccine is going to be better? So far, the RNAs look better. You know, the AstraZeneca wasn't as good. So Fauci takes 6,000 units a day. Yeah, you can actually go quite high with vitamin D. I just take the MDA. So does Daniel. But I think that would be fine. A lot of patients get back pain a week after COVID. I don't know what's causing it, but I assume it's inflammation related, right? I asked Michael Warby, and he said no. He said, I'm too busy, which is sad because you're not too busy to give us an hour, and a lot of people, like you listeners, appreciate it, right? But that's what he said. And thank you for the 2 thumbs. But maybe if more people bug him, he'll do it. Why is the Astrazeneca not getting media attention? He's not going anywhere in the U.S. It's being used elsewhere, but U.S. media is U.S. centric. Gotta wonder who the thumbs down. Well, there are always people that do thumbs down. It's fine. My virology class, I give a quiz to the students every 15 minutes, and some of them just pick the wrong answer to bug me. That's fine. That's fine. Epidemiologists versus virologists. They're not versus, actually. We have different viewpoints. The epidemiologists look at infection patterns, and they say we can tell what's going on with the virus, but they can't. The virologists can know what's going on with the virus, but we don't know about infection patterns. We both have to come together, but, the epidemiologists, the genomics people think they can conclude about transmission just from their patterns. You cannot. You cannot do that. Do you know of any research concerning infectious virus in different environments? So people have certainly looked in all these environments. And in fact, the upper atmosphere, there was a great paper published a couple of years ago, which we covered on TWIV, showing the viruses are whipped up in the oceans. They go into the upper atmosphere, and then they travel, and you can trace them by the genomics. But they're just looking at nucleic acids. They're not looking at infectious virus. Yeah. Are you planning any shows on prions? But not as long as we have SARS-CoV-2, which I don't think will be, as I said, maybe by this summer we can start thinking about having, or this spring, having other papers as well. Red mice don't have ACE2 in their lungs. Well, they must have some because the virus goes down and reproduces there. They have it in their upper tract. They get infected. They get mild disease. They don't get serious disease, though. That's for sure. All right, so I get this question all the time. Why can no one show proof of proper isolation? Of course it was shown. It was shown last year in January. I have the paper if you want to read it. For God's sakes, they put saline into a person's lung who had pneumonia. They pulled it out. They looked at it under the electron microscope. They saw coronavirus. They put it on cells. It grew. It stained with antibody to coronavirus, and they sequenced the genome. It's a coronavirus. What else do you want? You just didn't look at the paper. What are my thoughts on Moderna blocking transmission? Well, we don't know if it does. They were in the middle of doing a study, and they stopped because of lack of funding. So if you're asking me my opinion, I think it will block transmission or reduce it, okay? But we have to show it. Are there any viruses that don't mutate causing variants? Well, all viruses mutate. Whether the mutations mean anything is the question. Poliovirus mutates, but we've used the same vaccine for 70 years now against polio, right? Measles vaccine, we use the same vaccine since 1960s. The virus mutates, but none of the mutations affect vaccine immunity. And so, yes, all viruses mutate, but it doesn't matter for all of them. Oh, 2 thumbs up cancels the first. Okay. It makes sense. So 2 thumbs down will cancel it too, right? Dr. Fauci is using viral strains and variants interchangeably. No, that's not correct. But look, I am a stickler for nomenclature, and I understand he doesn't care. But a strain really means a certain thing. The virus has to have some new biological property. And I've noticed the mainstream press is using variants more and more because, out of the UK, they're called variants of concern, right? So that kind of does it. But strain is not right. You can always challenge me on virology, and I will tell you why I take my position. It's fine. I like being challenged because I can explain to you what's going on. It's a teaching moment, right? How about organic chemistry as a prereq? You don't need organic chemistry, and very little organic chemistry. Biology is better. Sorry you missed Amy. Come back next week. She'll be here. Your cholesterol went sky high post-infection. That's something I'd have to ask Daniel Griffin. Something to do with inflammation. Super spreaders. So the term has been changed. We call it super spreader events now, right? Where an infected person goes into a crowd and they infect a number of other people. So there's nothing special about them. It's just that they were in an event where they had contact with a lot of people. Okay, so they're not shedding more than average. Yeah, it's a gathering that infects more. That you got it, Aimweir. Perfect. Perfect. I'm not going to get the vaccine due to my health condition. Well, look, if you get infected, you could take monoclonals. That's for people who can't be vaccinated. That's what they're doing. Do you have any data on how the weather affects transmission, the environment? Well, yes, there's a lot. I don't have it myself. But, for example, I'll give you a couple examples. Warm weather increases mosquito range, and mosquitoes spread viruses. So mosquito range is actually moving northward as gets more warmer northward. Another example, respiratory viruses are better transmitted in the winter where it's cold. It keeps their infectivity longer, and low humidity, because when the humidity is low, the droplets that we expel travel farther. In the summer, when it's humid, the droplets absorb water and they fall to the ground. We need 9 more thumbs up to get us over 300. How long do I expect that vaccine-generated immunity will last? Well, we don't know. Let's say what we expect. I would say based on natural immunity to COVID infection, I think it would last a few years at least. But even if it lasted a year, getting immunized every year I think wouldn't be an issue. But I think it would be a couple of years. Does higher cholesterol affect the seriousness? I haven't heard that at all. No, I have not. But, if you have comorbidities like obesity, that can affect your immune responses. Well, thank you, David. I appreciate it. I just want to teach. In the end, I'm a teacher, and I enjoy it. And this is now these live streams are great because I can. Hey, thanks. You put me over 300. You guys are great. Can patients have long COVID potentially reinfect themselves and others? So there's no evidence that for the long COVID, there's no virus around. It's just the symptoms that are persisting. It doesn't seem to be a chronic infection unless you are immunosuppressed and have issues clearing it, but that's pretty rare. You watch all the TWIVs. Good for you, Pam. That's great. I love it. Doesn't matter if you didn't go to college. Although, yeah, I understand. It's good to learn stuff. Have there been any documented spillbacks to New World bats? I have not seen any, but we have not done a lot of wildlife sampling. So I'll bet it happens, frankly. We'll see. The WHO group going to China is going to be looking at that. If adenovector is used as a vector, does it make adeno dubious for a future vaccine? So that's a good question. Yes, I think if you get an Ad26 vectored vaccine, then you probably shouldn't get Ad26 vectored vaccine in the future. But there are enough different adenoviruses to get around that. That should not be an issue. We talked about colchicine earlier. Yes, it's being used. It's an anti-inflammatory, and that makes sense, as Amy said, because it's an inflammatory late disease for sure. Fauci wrote the book on internal medicine used in medical schools. All right. That's great. You think Fauci changes what he says based on what he thinks or the government thinks the public needs to hear in order to cooperate? I think many public health officials do that, yes. Is it valid to say more people infected, the more mutations? Yes. The more hosts, the more cells, the more reproduction cycles. Yes. More mutations for sure. All right. It's 9:30. Let me answer a couple more. Have I been vaccinated? Yes. I got my first dose 2 weeks ago, and I will hopefully get the second dose on the 16th of February. UC Berkeley is planning for classes in September. Yeah, I think that's reasonable. What happened to the instant test strips? They never got anywhere, sadly. Companies are having difficulty doing them. Novavax is in phase 3, about a month or two before the results are known. When will kids get the green light? In the U.S., we're thinking 2022. Because the vaccines have not been tested in kids or pregnant women. So you're really taking a risk to immunize them without making sure about it. What are your thoughts on Moderna blocking transmission? I think it will. I'm just hesitant because we don't have the data yet, and I'm a scientist. I'm driven by data, but I think it will block transmission. Okay, folks, I'm going to say goodnight to all of you. And it's been a pleasure. I think you're great. You have great questions. Every one of them are great questions. And I love answering them. So thanks for coming. And we'll see you next time.